chapter on strategies for enhancement in food production, you have already studied animal husbandry. Today, we will focus on plant breeding, single cell proteins and tissue culture. But before we start with this topic, it is important to understand the difference between wild, cultivated and domesticated plants. Wild plants are those plants that grow unchecked in natural environment without any interference from human beings. Now, this slide shows foxglove growing in the wild along with other wild plants. Cultivated plants are those plants that are intentionally selected by man for cultivation for his use, like for example azaleas, which are a subgenus of rhododendron. Then we come to domesticated plants. Domesticated plants are those plants that live in close association with human beings. Domestication is genetic modification, that is in a way controlled breeding, for example maize and bread wheat. This is maize field and this maize is dependent for its survival on human beings. In animals also, I can give you an example to understand the word domesticated properly. Dog is a domesticated animal and it cannot survive without human help. Now we come to wheat, which was and which you know is the first cereal crop to be domesticated by man 12,000 years ago in the Fertile Crescent in the Middle East. Now, what is this Fertile Crescent? Where is it? Fertile Crescent included eastern Turkey, Iraq and southwest Iran. Agriculture began 10,000 to 15,000 years ago in 11 different centers around the world. Classical plant breeding involves crossing or hybridization of pure lines, followed by artificial selection to produce plants with desirable characteristics of yield, nutrition and resistance to disease. Today, molecular genetic tools are used in plant breeding to incorporate traits like crop yield, improved quality, increased tolerance to environmental stress like salinity, extreme temperatures, drought resistance to pathogens like virus, fungi and bacteria. Increased tolerance to insect pest is how is it done? Breeding a new genetic variety of crop requires the following steps. First, collection of variability or germplasm. Second, evaluation and selection of parents. Then, cross hybridization among selected parents. Selection and testing of superior recombinants. Then, and the last step is testing, release and commercialization of new cultivars. So, we discuss the first one, collection of germplasm. Many crop plants have genetic variability from their wild relatives. Therefore, it is important to collect and preserve all wild varieties, species and relatives of the cultivated species. The characteristic of these plants is evaluated and the complete collection of plants or seeds with diverse alleles for all genes in a given crop is called germplasm collection. Then you have evaluation and selection of parents, and the germplasm is evaluated to identify plants with desirable characteristics. The selected plants are multiplied and used in the process of hybridization. If possible, pure lines are also created. And third, cross hybridization among the selected parents. Now, supposing we have selected two parents, A and B. Plant A has high protein quality and plant B has disease resistance. For obtaining a hybrid AB with both these characteristics, we have to hand pollinate the stigma of B with the pollen grains of plant A or vice versa. It is a gamble. The hybrid may or may not have the desirable traits. Generally, only one in a few hundred to a few thousand crosses show the desirable combination. And fourth, selection and testing of superior recombinants. After hybridization of parents A and B, we have to select among the progeny of hybrids those plants that have desirable characteristics. For example, in AB hybrid, we want both high protein quality and disease resistance. These are self-pollinated for several generations till they reach a stage of uniformity, that is homozygosity. This is done to prevent segregation in the progeny. And then fifth point, testing, release and commercialization of new cultivars. The new hybrid plants or selected lines are evaluated for the yield and other agronomic traits of quality, disease, resistance, etc. The plants are grown in research fields under ideal fertilizer application, irrigation and other crop management practices. If the plants grow successfully in the research field, they are tested in the farmer's field for at least 3 growing seasons at various locations in the country, that is in different agroclimatic zones. It is evaluated in comparison to the local crop cultivar. This photograph shows some Indian hybrids, maize, wheat, garden peas, and this is a protocol given by Professor K.R. Shivanna and Professor Sahani. Both were head of the departments earlier in Department of Botany, University of Delhi, and this protocol is for plant breeding between cultivated plants and wild plants. And in March 1968, Director William Gord of the US Agency for International Development, USAID, first used the phrase green revolution to describe the great gains in field of rice, wheat, maize and other crops through the use of high yielding variety. And similarly, in rice, dwarf and semi-dwarf varieties were released, which increased its production manyfold. Anxious at the growing gap between the rates of growth population of food production, India's first Prime Minister Jawaharlal Nehru said in 1948, "Everything else can wait, but not agriculture. India is one of the original centers of rice. Now, at the time of India's independence in 1947, the country produced about 6 million tons of wheat, and through these high yielding variety program HYVP, wheat production rose to nearly 17 million tons in 1968. To commemorate this great achievement, the then Prime Minister Indira Gandhi released a special stamp titled Wheat Revolution in July 1968. India has released several postage stamps depicting crop plants. These are some stamps of wheat and rice, where you can see on the left women planting rice, and on the right man with the wheat straw, and in the center the World Food Day. Now, some of the wheat varieties that were developed at IARI were Kalyan Sona, Sonalika, Sharbati, Shonara. Dr. Norman Borlaug, the father of Green Revolution, was awarded Nobel Peace Prize in 1970 for his outstanding contribution to humanity. He was able to introduce Mexican, semi-dwarf and dwarf varieties Shonara 63, Shonara 64, Lerma Roja 64A, all of which are bred wheats. Dr. M.S. Swaminathan, an outstanding geneticist and grain revolutionist, also former Director General of International Rice Research Institute, Manila, Philippines, developed Sharbati Shonora variety and was responsible for starting an era of dwarf wheat varieties in India. The top 10 rice producers in the world, according to data gathered by the International Rice Research Institute in 2009 are: first is China. China produced about 193 million metric tons, and that is 35% of world production, and China is the world leader in hybrid rice production. Second is India, then Indonesia, Bangladesh, Vietnam, Thailand, Myanmar, Japan and Philippines, Brazil. Now, the top crop producing states of India. The largest producer of wheat is Uttar Pradesh. Rice is in West Bengal. Largest producer of maize is Andhra Pradesh. The largest producer of coarse cereals is Karnataka and Rajasthan. Now, we will discuss about Saccharum officinarum, that is noble cane or sugar cane. Now, this species of sugar cane has soft, thick stemmed, large barrel with low fiber and high sucrose content. But it is important to note here, it is susceptible to almost all the serious diseases that have been reported for the crop. Then you have another species of Saccharum barberi, Indian cane. It is intermediate between wild and noble canes. It has hard, small barreled stem with high fiber content and has a significant amount of juice, and is fairly resistant to some diseases like root rot and mosaic. Crosses were made between noble cane and Indian cane to produce resistant varieties. Also, noble cane was crossed with wild types like Saccharum spontanum to produce hybrids that combine the best features of both parents, that is thick, soft, long barreled cane and high sucrose content and deep penetrating root system, vigor, drought and disease resistance of the wild parent. Breeding work in maize, jowar and bajra have also produced several high yielding varieties resistant to water stress. Now, what are the uses of plant breeding? Plant breeding is done for raising disease resistant plants which are resistant from fungal, bacterial and viral diseases and other pathogens. This is a table showing how plants have been raised which are resistant to various insects and various pests. Mutations can also be induced artificially by using chemicals or radiations like gamma radiations and selecting and using plants with desirable character as a source in breeding. This is known as mutation breeding. Plant breeding in developing resistance to insect pests, and that table shows crop plants and various insects which attack them, and hybrids have been raised so that they become resistant to those pests. To prevent crop destruction, insect resistance can sometimes be achieved by selecting morphological, biochemical or physiological characteristics. Generally, hairy leaves are associated with greater insect resistance. For example, resistance to jacids in cotton and cereal leaf beetle in wheat. Smooth leaved nectarless cotton varieties do not attract bollworms. Third, in maize, resistance to maize stem borer can be achieved by selecting a variety with high aspartic acid, low nitrogen and sugar content. Plant breeding is also done to improve food quality. Millions of people in the world do not get enough food to meet their nutritional requirement. Many suffer from micronutrients, protein and vitamin deficiencies. Diets lacking in essential micronutrients, particularly iron, vitamin, iodine and zinc, increase the risk of disease, reduced lifespan, reduced mental abilities, and therefore, what we do is biofortification is done, and biofortification is breeding crops with increased levels of vitamins and minerals, higher protein and healthier fats. Now, what are the objectives of biofortification in a little detail? Breeding for improved nutritional quality is done with the aim of improving protein content and quality, oil content and quality, vitamin content and micronutrient and mineral content, for instance. Now, in 2000, maize hybrids were raised with twice the amount of amino acids, lysine and tryptophan. Wheat variety Atlas 66 with high protein content was used as a donor for improving cultivated wheat. An iron fortified rice variety with 5 times more iron than local varieties was developed. Similarly, vegetable crops that are rich in vitamins and minerals were released by IARI PUSA. These include pumpkin, carrots, spinach, and they were enriched with vitamin A, karela, bathua, mustard, tomato, enriched with vitamin C, spinach and bathua enriched with iron and calcium, legumes of course enriched with proteins. Now, this photograph shows a hybrid papaya. This is hybrid papaya, and similarly many hybrids have been raised. Now, single cell proteins, SCP. Single cell proteins are derived from a culture of edible single celled organisms, and these are an alternate source of proteins for human and animal nutrition. The protein extract can be obtained from pure or mixed cultures of algae, yeast, fungi or bacteria. Microbes employed in single cell proteins are chlorella, spirulina, other blue-green algae, this is microalgae, and first spirulina is the world's first superfood. Like then you have yeast, and similarly a number of other or microorganisms can be used. Also fungi, Aspergillus fusarium, these can be used for obtaining single cell protein. Then we come to plant tissue culture. Plant tissue culture is a technique that can be used for growing and maintaining plants, cells, tissues or organs under sterile conditions on a nutrient medium of known composition. These are also called in vitro cultures. The part of the plant used for raising the culture is called an explant. There are different types of explants. Meristems can be used, phloem cells, parenchyma tissues like endosperm, nucellus, and organs like ovule, anther, they can be used for raising tissue culture plants, and these plants can enhance plant production of many economically important plants. Now, to go in a little detail about the explants, these explants can be obtained from portions of stems, leaves, roots, flower buds, inflorescence, ovule. From the ovule you can take out the ovary, nucellus, endosperm, embryo, and from the anther you can culture the whole anther or your microspore mother cells, pollen grains can be taken out from the anther. Then you can do meristem culture, and then protoplast culture can also be done. This photograph shows ovule and nucellus culture of cucurbits and cotton. The ability of a cell to regenerate a whole plant is known as totipotency. This was first demonstrated by F.C. Stewart way back in 1930s in carrot. He was able to raise a complete plant from a root cell. These can be used, carrot has been used, similarly many other plants have been used for plant propagation through tissue culture. And advantages of this tissue culture techniques are numerous. First, plants produced through tissue culture are exact copies of the parent plant. Virus-free plants can be raised through meristem culture, example in cassava, potato, sugarcane. Third, the technique of tissue culture can be used for propagation of rare and endangered plants like Brahmkamal from Uttarakhand. Whole plants can be generated from plants that have been genetically modified. This technique helps in generating transgenic plants also. Now, plants grown in vitro can be shifted from one place or one country to another country without any fear of transmitting diseases, pests and pathogens. In those plants where seed germination is low, tissue culture can be done, and example of such plants are orchids, then Nepenthes. These can easily be done. A lot of work has been done on orchids, meristem culture, and this is a photograph of orchid, and in orchids meristem culture has been done to raise virus-free plants, and virus-free plants can be raised from any plant because the meristem, once the explant is taken from the shoot tip, is without virus. This technique can also be used for raising somatic hybrids or parasexual hybrids. For example, the protoplast of tomato can be fused with the protoplast of potato. The hybrid protoplast first develops a cell wall and then undergoes divisions to form a mass of cells and ultimately differentiates into a plant. The hybrid plant is called, in this case, pomato. But this pomato was not successful commercially because it did not have the desired characteristics. Parasexual hybrids have been raised in a number of plants. For example, in citrus, brassica and in many other important plants. To conclude, plant breeding is a technique for creating varieties that are resistant to pathogens, insects and other diseases. To improve the quality of food, especially protein content. Single cell protein SCP is an alternate source of protein obtained from chlorella, spirulina, and we have technique of tissue culture and somatic hybridization, which offer vast potential for manipulation of plants in vitro. Thank you.